Welcome back. Okay, today's lesson is going to be more about the controlled experiment. The controlled experiment is a super important part of scientific research. It's basically common sense, though. I mean, it's not super exotic. It just has to be very conscious of this is how it's proceeding in order to be valid. Um, all it really amounts to is a fair test. If you want to find the effect something has, you have to be able to compare it to a situation where it wasn't there. Now, I'm going to tell you one situation right now that's happening at this moment that's really super important for all of us is the development of a vaccine for COVID, right? And right now, they're in what's called phase 3 trials of COVID vaccines that are being developed. Phase 1 and 2 would be developing something that they're testing. Okay, is it seem like it's safe with a small group of people, or and also does it generate what's typically an immune response, making antibodies? Well, they said, wow, it seems to do that. Now they need to find out, does it actually prevent people from getting COVID? Vaccination is a very well established, uh, type of a treatment to prevent certain kinds of diseases like smallpox and things like that. And so they're trying to develop one for COVID, but it's got to be tested to find out if it works. And so the way they do it, they have one group of people that get the vaccine, and other group of people, and at this point it's always got to be volunteers, and at this group of people where they get a placebo. They don't get the vaccine. They still get the shot or whatever. They don't know if they have the vaccine or not. And then you study them over time and you see, is there a difference between this group of people that got the vaccine getting infected or not getting infected and the ones that did not get the vaccine? Did then more of them get infected? So what we're hoping is that the infection rate on the people that didn't get it would be as if there's no vaccine, but the people that got the vaccine would not be getting COVID. And that would make it a successful test. And the safety also is being tested if there's a lot of side effects or something of that sort. So this is a very important thing right now for all of us. A lot of people believe that's what's going to help us get back into the classroom is the development of a vaccine. So scientific research, depending on the controlled experiment, or in this case, what they call it is a randomized clinical trial. Randomized means that the people included in the group are picked at random, and some are given the treatment, and some are given a placebo, and to see if there's a difference. It has to be randomized because you don't want to be giving it. Oh, we're going to give the vaccine just to healthy people and not to others. Well, that would influence it. So those all the factors, all the variables have to be evened out so it's identical situation. So there's a vocabulary here for this. And so what you need now for you to know the three different types of variables there are when you're talking about a scientific study. Okay, and the vocabulary words for them, I will tell you, when they first started doing the standardized testing in Washington State in the year 2000, I've been seeing this going on since then, and they always have these vocabulary words in a standardized testing. When in 8th grade, you'll have to take the science test. And so the variables lesson. Here we go. So please write down on your paper variables lessons. This is the assignment we have. And boom. First vocabulary word, manipulated variable. Write that down. Manipulated variable. Now, what the manipulated variable is, that's the thing that scientists are trying to test. They're trying to find out if it has an effect. For example, a vaccine or a headache remedy or whatever it is. It could be like the manipulated variable. If you're trying to find out, oh, I don't know, if different balls bounce the same height or something, it's like, well, the difference would be the balls, right? And so those would be the manipulated variable. So whatever it is you're actually trying to ask the question about to see if it has an effect on something else. Okay? And so write the definition down. Boom. This is what factor the scientist changes in an experiment to see if it affects the system. So you have two different groups. You have the group that gets the vaccine, the group that doesn't get the vaccine. What's different, what has changed, is the presence of the vaccine. Or what's changed is the type of ball that's being used. I probably shouldn't use a green ball with a green screen. Maybe it's an invisible ball. That would be funny. Oh, well, whatever. Anyway, the manipulated variable, you can think of it in terms of that's the one that the scientist manipulates. he changes as he does the experiment, like whether you get the headache medication or whatever it is you're trying to test. Okay, so you're trying to see if it has an effect on the system. That's the manipulated variable. Moving into the next one is the responding variable. What you want to see if this one responds to the presence of the manipulated variable. For example, does the infection rate for COVID respond to the presence of the vaccine? Does the vaccine prevent infection? So that would be, does the infection rate respond to the presence of the vaccine? So this is what you're measuring. This is the data that you're taking. If you're doing it for different kinds of balls, dropping and how high they are, well, what your responding variable is the height that they bounce, if that's what you're measuring. If you're measuring like different paper towels, how absorbent are they? Well, the responding variable would be the, I don't know, how much water they absorb. And so responding variables, that's the measurement you take. That's the data you get, right? And you use that to determine if the manipulated variable has an effect, if there's a response. By the way, and this is kind of just a troublesome thing, there's two different words for this. There's the manipulated and responding variables. That's what we say in Washington State, but kind of a little more widespread than that. There's the independent and dependent variable. It's the same exact thing. The independent variable is the manipulated variable, and you want to see if the dependent variable depends on what happens in the independent variable. So the responding variable is also known as the dependent variable. Okay, I know that. Okay, so the responding variable, write this definition down. This is what factor or factors the scientist measures in an experiment to see if changing the manipulated variable affected the system. So this is the measurement you take. So go ahead, write that down. Now you might want to pause the video and write it down. I'm going to go ahead and move on with my lesson. Okay, so what factors the scientist measures in a system? Now the controlled variables. Now this is all the variables that have to stay the same in order for it to be a fair test. That's what it means to be a controlled experiment. It doesn't mean, oh, you're being really careful or you're controlling what the data will be. No, it doesn't mean that. What it means is you have the controls, which are what you can compare it with. For example, the experimental group is the one that, say, in this case I used before, the fertilizer X is sprayed. Well, the control group is where it's not sprayed. All the things are equal except for whether it has the fertilizer. All the variables, such as how much water you give it, how much light it has, the temperature, the amount of soil, the kind of plant, the health of the plant, everything is equal. Those are controlled variables. And when you're doing a clinical trial, like they are for right now with the vaccine, they do that controlled variables by randomizing who gets put in which group. You don't get to choose, hey, I want to be in the group that gets the vaccine. You don't get to choose. You don't know which group you're in. And again, it's all explained to a person that's involved in that. They understand you may be getting it, you may not be getting it, because that's what makes it a fair test. Because then you are, there's should be, if you have enough people and they're randomly selected, there should be no difference statistically, one group versus the other. Please write this definition down. These are what factors the experiment set up that the scientist must keep the same. The experimental setup is how it is. Everything has to be identical to make sure the manipulated variable is the only thing affecting the responding variable. You want to try to sort of isolate the impact of the manipulated variable, to isolate its influence, to see if there is influence. And if it turns out that there's no measurable statistical difference between the two groups, then you realize it really didn't have an influence, right? Okay, so this is the classic method scientists use in their experiments to find out what effect one thing has on another. Okay, so in review, you have the manipulated variable, and that's also known as the independent variable. And this is what factor the scientist changes in an experiment to see if it affects the system. You have the responding variables. These are also known as the dependent variable. And these are what factors the scientist measures in an experiment. And by the way, this is written in yellow, so you don't have to write it down again, okay? the scientist measures in an experiment to see if changing the manipulated variable affected the system. And then the third one is the controlled variables. And these are what factors of the experimental setup that the scientist must keep the same, to make sure the manipulated variable is the only thing affecting the responding variables. So manipulated variables, also known as independent, is what scientist changes. Responding is what he measures, and controlled is what she keeps the same when she's doing the experiment. All the factors that have to be identical. Those are the three variables. Okay.